Yeah, so today we'll be starting with periodontics. Okay, now when we talk about this subject, whenever we talk about any clinical subject rather, the problem that we have is they are vast, they are huge. But advantage that we have is they are easy, as far as with the fact that we have read them very recently, especially the ones who are interns, they have read these subjects in their final year, and we have read them in more details and in a more better way. So that's one advantage. But disadvantage, yes, it is, it's gonna be very vast. Okay, so how do we deal with it? Since you have read it and since you are accustomed to it, we may move a little bit faster as compared to the way we deal with basics. Okay, anyways, still the things are in your hands, so anytime you feel things are not clear, I'll be there to slow down and explain and all those things. But be with me, be concentrate and try to move as fast as possible, because the basic aim is, let's spend as little time as possible per subject and get the most out of it. Okay, so in today's class I'll try to deal with gingiva, PDL, and the basic part, the structure of periodontium, and if the time permits I'll be going into plaque calculus, the etiology part. We'll be starting with that thing. Okay, all of you have that problem? Usually interrupted audio or video has two things. One is low bandwidth from my end or low bandwidth from your end. If it is low for my end, it will be bad for everyone. Okay, if it is low from your end, that's something like it will be only for you, it will be bad, and that's where I'll not be able to do anything. It's the internet speed problem. Okay, so hopefully, usually, close everything else, close any other window, any downloads or anything. If you have the speed well, that should work fine, right? Let's see, Arnab, how it holds on for you. Okay, so are we good to start? Yeah, in the beginning we'll have people logging in, logging out and all that, a little bit distractions, but should be fine. Okay, so when we talk about gingiva, of course we always start with oral mucosa. Now we all very well know, histology point of view, our oral mucosa is divided into three types: the masticatory mucosa, the specialized mucosa, and the lining mucosa. Masticatory, the hard palate and gingiva, wherein we have the lining of keratinized epithelium, which is very much adapted to bear the stress. Specialized has some specialized function, like we have in tongue. We have a special function of taste. Then we have the lining mucosa, wherein, all the rest of the cavity, wherein we do have the epithelium, but it's usually non-keratinized and not so adapted to stress. Right, so you all know these things, nothing great in that. Still let the chat box be ticking, as so that I know that I can move on quickly with these things. Okay, now the term periodontium, when I say the term periodontium, it consists of gingiva, PDL, alveolar bone and cementum. Okay, but when I say the term periodontal ligament, the term periodontal ligament itself just means the periodontal ligament, that is between cementum and the bone. But the term periodontium, it includes all of these, the gingiva, the PDL, the bone and the cementum. The whole apparatus is called as the periodontium. So that is one thing you do have to notice when it comes as, in the sentence as MCQs, although it's not much significant. Let's move on to gingiva. Now when we talk about gingiva's division, now look at this diagram. Structurally, a gingiva is divided into marginal, attached and interdental gingiva. Okay, so now marginal gingiva is, if you try to insert your probe, there will be this end after which the probe doesn't go in. This is the sulcus, the depth of the sulcus. From the depth of the sulcus to the tip of the gingiva, this part of gingiva is the marginal gingiva, right? So after that is your attached gingiva. After your attached gingiva, at a particular point, your attached gingiva ends. That end is the mucogingival junction. After that, you have your alveolar mucosa. That is a lining mucosa. You know, when I say alveolar mucosa, it's a variety of lining mucosa. We all very well know that lining mucosa is found in all the other regions of oral cavity, including the buccal mucosa and all that. When we say alveolar, it is the lining mucosa that covers the alveolus. You know, so this is attached and this is alveolar mucosa. Now, we'll be actually seeing further where we have what kind of epithelium, you know, as we move on with the slides. But at this point itself, let me highlight one difference. If I say keratinized gingiva, okay, if I say attached gingiva, just imagine the two terms. Do you think they are just the same? The width of which gingiva will be more? Will it be the keratinized gingiva that will be more in width, or will it be the attached gingiva that will be more in width? Yeah, now let me explain the things. Okay, now this is our marginal gingiva. From the end of the marginal gingiva to the beginning of the mucogingival junction is our attached gingiva. You all understand this part, this definition of attached gingiva. After that, we have our lining mucosa started. But now when we talk about the keratinized gingiva, you know, out of whole these gingiva, the marginal and the attached gingiva is keratinized. And alveolar mucosa is non-keratinized. So when we talk about the keratinized, it will include the marginal gingiva and the attached gingiva to be called as the keratinized gingiva. Get the point? So which is more? Good, yes, it's a keratinized gingiva. That is more in width. An attached gingiva is a part of keratinized gingiva, while marginal gingiva is the remaining part of it. Okay, so hope you get that difference, and with that we move on to see individual gingiva, that is free gingiva and all that. Now in the last slide we saw the marginal gingiva. Let me talk about one more thing. Okay, here we saw the marginal gingiva, and there is one demarcation which we can see, you know, which can differentiate between marginal and the attached gingiva. This demarcation is called as free gingival groove. Okay, now this free gingival groove, there are a lot of hypotheses why we have this free gingival groove. Now, we all very well know that marginal gingiva is keratinized and attached gingiva is keratinized. What is the difference between the marginal and attached gingiva? That the marginal gingiva's end, there is non-keratinized gingiva here, Saurabh, in alveolar mucosa. There is non-keratinized gingiva in the sulcular epithelium, inside here. But when we talk about the outer side of the marginal gingiva and when we talk about the attached gingiva, they are keratinized. Okay? Yeah. Now, as I was talking about this free gingival groove, this free gingival groove is a representation of the depth of the sulcus. So if we say our depth of the sulcus is here, you know, a free gingival groove will be an external manifestation. So inside, wherever our depth of sulcus will lie, if you project a line outside on the outer surface, you get the free gingival groove. So that's how it, you know, you can actually arbitrarily say that this should be the depth of the sulcus inside. Now this point is not very strictly applied when we see it clinically, and there's one more point that we don't see free gingival groove you know, in all the patients. Okay, so yeah, now look at this point over here. The free gingiva or the marginal gingiva is separated from attached gingiva by free gingival groove in 50% of cases. right? Rest of the 50% of cases, what do we say? That doesn't mean that they don't have marginal gingiva. That simply means that there is no, obvious free gingival groove, right? So what is free gingival groove? It is nothing but the external manifestation of the depth of the gingival sulcus. Okay, now gingival sulcus, when we talk about the depth of the gingival sulcus, in very ideal conditions, in the gnotobiotic animals or the animals which are microbiologically sterile, in ideal condition we should have this sulcus as 0. But that's not the scene. That doesn't mean, we consider ideal as healthy. Clinical healthy definition is something else. Clinically healthy definition is, wherein we just rule out the disease by some other criteria, and in those conditions the depth is 2 to 3 millimeter when we talk about the probing depth. Okay, and when we talk about the histological depth, it is 1.8 millimeters. Now at this point, let's talk about what is histological depth and what is probing depth. Well, things are quite obvious when we probe, when we put a probe to measure a sulcus. Okay, that depth, whatever we get, is your probing depth. But if you actually take a section and make a histological slide, so you will actually be able to locate the, this end, the upper end of the junctional epithelium, and from there when you measure the sulcus in the histological slide, that will be 1.8 millimeters. The range is 0 to 6 millimeters. Now, why do we have that difference between the probing depth and the histological depth? Because probing depth depends on many things, especially it depends on the force. And most commonly, when we probe, we actually go a little bit inside the junctional epithelium. That is the reason we have our probing depth more than the histological depth, right? Get this point? Now one thing, your marginal gingiva, as we defined it, it is the one which has the sulcus. So obviously that is the free gingiva, marginal free gingiva, and it can be separated from the tooth by periodontal probe. That's obvious point. Nothing great in that. Clear till now? Now let me tell you one thing. All these simple points, all these, the points that appear very insignificant, they have a lot of questions. Gingiva takes a lot of share of MCQs from perio. Okay, even you can say this part is the histological part as well. So your oral histology will also have questions from this part. So you can say it's an overlapping part, and usually the overlapping parts are important. So this part is a very important part. Okay, so keep noticing all the small points. Now let's talk about attached gingiva. Now you all very well know from where to where we had our attached gingiva. We had it from the free gingival groove to the mucogingival junction. That is, from the point where our marginal gingiva ended, our attached gingiva started, and then it continued in the mucogingival junction from where our alveolar mucosa started, right? So that is our attached gingiva. Now, very common questions about attached gingiva is the width, which area we do have the maximum width. It is greatest in the incisor region. So what happens is, when we actually look in the book says the thickness, the width of attached gingiva is greatest in incisor regions, and then it gives all these things in kind of brackets and all that. So if you notice, it's 3.5 to 4.5 in maxilla, okay, 3.3 to 3.9 mm in mandible. Now if I frame my MCQ, width of attached gingiva is maximum in, and then the options are maxillary incisors, mandibular incisors. What would be the choice? Rashmi, you have the question and the choices over there. It's like, which is greater? 4.5 is greater or 3.9? This is fill in the blank. You have to put less than, equal to, or less than 5. Okay? This one. So this is a simple mathematics over here. Why I am talking about this point? Because there is an MCQ that talks about Okay, now let's talk about the least amount of attached gingiva. We have least amount of attached gingiva in the first premolar area. Now look at the values. The value says it is 1.9 mm in maxillary premolars and 1.8 mm in mandibular premolars. So where is your attached gingiva least? It is mandibular premolars. Your attached gingiva is least. Okay, and I'm amazed after having such a direct reference and direct values. This question is a very highly discussed question on the forums. Okay, hopefully I will not see you all discussing this question. So all clear till now? Yeah. Then we talk about the stippling. Now as we come to attached gingiva, the attached gingiva is the one that is attached to the underlying bone. Now this attached gingiva has a very special feature of stippling. Why it has stippling? The mucosa of this attached gingiva shows these, the epithelium shows these retipegs which are kind of intertwined with the projections from the underlying connective tissue. So because of this we see stippling in attached gingiva. Now the next point is the width of attached gingiva. It increases with age and in supraeruptive teeth. Again, this is the one thing wherein people say with age we have recession, then how do width of attached gingiva increases with age? Now let me tell you one thing. Recession is pathology. It's not a normal aging changes. Right? So always understand one thing. When we say recession, it's not something normal. That, although the point that with more age, there are more cases or more people having recession. But that doesn't make recession normal. Get the point? Okay, let me talk when usually, let me just go to the whiteboard. Yeah. So this is our epithelium. Okay. These projections of the epithelium downwards, they are called as retipegs. Okay. Now these retipegs have these connective tissue papillae going inside. These are called as connective tissue papillae. Get the point? Yeah. Now I was talking about width of attached gingiva increasing with age. Now we had been seeing the free gingival groove, and after that we had been seeing the attached gingiva. Now let me tell you one thing. Tooth has a very significant property that if the tooth erupts slowly, okay, if we try to pull out the tooth slowly, be it orthodontically, be it by some natural process, it takes the whole attachment apparatus with it. That means it takes the gingival attachment, that is the the junctional epithelium. It takes the bone, it takes the everything with it. Everything grows with it. Now there is one more point we always talk about is mucogingival junction. Mucogingival junction is genetically determined and it is stationary throughout the life, isn't it? That's what we talk about. Right? If the mucogingival junction is kept stationary and somehow if we are able to pull the rest of the attachment apparatus upwards, what will happen to the attached gingiva? The attached gingiva will increase, isn't it? That's what is the explanation for increased width of attached gingiva with aging. What happens as you age, there is attrition. Now this attrition is a normal phenomenon, the physiological phenomenon. It's not considered pathology. Now because of the attrition, what happens is the occlusal surface is lost. Now the tooth goes out of occlusion. The tooth gets a chance to supraerupt. Now the tooth supra-erupts. When it supra-erupts, there is no recession. What exactly happens is, it takes the gingiva along with it. It takes the whole attachment apparatus along with it, and that moves your free gingival groove upwards, increasing the width of attached gingiva. So width of attached gingiva increases with age. Width is measured from here to here. Free gingival groove to mucogingival junction. I understand. Mathematically, when we talk about this, we'll call as height, and this we'll call as width. But no, its width is from free gingival groove to the mucogingival junction. Okay, all clear till now? Yes. So as we saw, your mucogingival junction is the one that separates the attached gingiva from the lining gingiva. So mucogingival junction, we can actually demonstrate this mucogingival junction by Schiller's iodine. And this Schiller's iodine actually stains the lining mucosa, because lining mucosa has glycogen and it stains the lining mucosa darker, so we can actually see the attached gingiva against it. Okay, now mucogingival junction is stable with increasing age, at least in relation with nasal floor and the lower border of the mandible. Now whenever we say this particular thing is stable, how do we say it is stable? When we take some other landmark, and in relation to that particular landmark, if this thing is not moving, then we say it's stable, right? Because stability is a relative term, isn't it? We find the earth stationary, not moving, because we don't feel it, because we are moving with it, isn't it? Anyways, hopefully you get that point. Now in this diagram, you actually see the mucosa stained over here, and this is the lighter stained attached gingiva. And because the lining mucosa has a glycogen that gets stained by the Schiller's iodine, and that's what helps us demarcate the mucogingival junction. Okay, done guys? Do you remember the width of attached gingiva in maxillary premolar area? Can you all be quick and write it in the chat box? Let's see how many of you. Maxillary premolar area. I'm getting 2 answers. It was 1.9 mm. And how much it was in mandibular? Mandibular was 1.8. Yes. And what about the width of attached gingiva in the maxillary incisor area? Okay, and how much it was in mandibular incisors area? Aparna, we need the whole range. Yeah, it was 3.3 to 3.9 millimeters. So as you move on with any particular subject, please look into the values that you have come through, because the values are the important source of MCQ. How much was the histological depth of sulcus in clinically healthy gingiva? See my question? I asked about histological depth of sulcus. Isn't it, Saurabh? That's where the mistake comes actually. Be very careful what is the question about. Okay? Yes, the clinical depth in a clinically healthy gingiva, or probing depth in a clinically healthy gingiva is 2 to 3 millimeters. Right? Okay, let's come down to interdental gingiva. Interdental gingiva is pyramidal in anterior teeth and cool shaped. Now look at this point. In anterior teeth, what happens? You have a pinpoint contact area, and just below that you have your interdental gingiva, so that looks like a pyramidal shape. But when it comes to the posterior teeth, we have a bigger area, and because of that you get this kind of a shape, which we call as cone. So this particular concave area is called as cone, and this particular shape is called as the tent shape, but in many books it will be just said as caul shaped. Okay, so caul shape or tent shape. Herein we have the pyramidal shape. Okay, now yeah, we have two pictures for incisors that just tells you, you know, if most of the times incisors have at the top, but many a times when the contact moves downwards, you know, if the contact moves downwards, the tip gets flattened. Basically, you know, your interdental gingiva will adapt the shape of the contact area. Okay, when we talk about your MCQ point of view, I would not actually want you to put your brains in those particular points right now. Just focus that this has a pyramidal shape and this has a tent shape. We see caul in posteriors because it is a broader contact area. Now, if somebody asks you a question that we shift the contact point of an incisor downwards, make it a broad shaped, what will be the change in the interdental gingiva that you expect? Now that's a common sense. If your if your contact area becomes broad and it has moved down, we will expect that this new contact area will result the shape of interdental gingiva to become like a caul shaped, like the way we have in posteriors, isn't it? So that's what it explains. So hopefully you understand this point over here, and this one point, this one. Let me explain you that. Lateral and mesial border of the facial interdental gingiva. When we say facial interdental gingiva, we are looking at the interdental gingiva from the front. When we look at the interdental gingiva from the front, now look at this. We have the free gingival groove running like this, isn't it? So we have our marginal gingiva running like this over here. So our marginal gingiva are making the sides of our interdental gingiva, and the center of the interdental gingiva is made up of attached gingiva. See that? See that point? That is why, remember, if in your Viva question in your final years, your professor will be very fond of asking. Yeah, what I mean to say is your marginal gingiva, which is going along, you know, it extends into the interdental area also. So in interdental area, if we consider this particular area, this is an interdental gingiva. Now, interdental gingiva is made up of marginal gingiva on either sides. You see this? This is a marginal gingiva on both the sides. But the center of the interdental gingiva will be made up of attached gingiva. Okay? Yeah. So the center of, so you can say, if I talk about stippling, where will you see stippling in interdental gingiva? In the borders or in the center? In the center. Because Sora, borders of free gingiva, we will not see stippling there. We will see it in the center. Isn't it? Because we saw stippling only in the attached gingiva, isn't it? So an attached gingiva present only in the center of the interdental gingiva. Okay? Clear, all of you? Kamal, Priya, Shrisha? Okay, so this what is about the interdental gingiva. Now, there is one point. Interdental gingiva will be kind of absent, or papilla will be kind of absent, wherein we don't have the contact point. Because when we have the contact point, when the teeth are closer enough, that particular space, look at this point. The teeth are close enough and we have the space here. This space is filled in by the papilla, but when the teeth are apart, there is no contact point and we do not have the papilla rising up there. So there is absence of interdental papilla and caul where the proximal tooth contact is missing. Now this is an important MCQ. Okay, this is a normal scenario wherein you can have a missing IDP. Clear with that? Yeah. Now let's talk about the microscopic variation of the gingiva. Now till now what we saw was macroscopic classification. Now when we talk about the microscopically attached gingiva, any gingiva is made up of stratified squamous epithelium. So your gingiva basically has squamous cells. Now you all must be knowing what squamous cells are. They are the flat cells, and stratified, that is they are in layers. Now when we differentiate what cells we find in gingiva, we have two basic classes of cells. One main class is the keratinocytes, okay, and the second main class is non-keratinocytes. So what do we see in gingival epithelium? We see keratinocytes and we see non-keratinocytes. Keratinocytes are those principal cells which actually make up the epithelium, the epithelial cells, you can say, okay. So those are our keratinocytes, and any other cell other than those keratinocytes are non-keratinocytes. So what do we have in non-keratinocytes? In that we have the melanocytes, we have the Merkel cells, and we have the Langerhans cells. Are you all with me? Now, melanocytes are present in the basal and the spinosum layer and synthesize melanin. Now, before we actually talk about which layer these cells are present, we should know the layers of the cells. So let me just go to this diagram. Okay. Now, whenever we have an epithelium, a stratified epithelium, in a stratified epithelium, we will have cells arranged in layers. Okay. So when we talk about a keratinized version, okay, a keratinized epithelium will have the layers like the basal layer. The basal layer is the one that is sitting on the basement membrane. Then we have the prickle cell layer. Prickle cell layer, also called as spinous layer. Okay. After that, we'll have the granular layer. Okay. After the granular layer, we have a keratinized surface layer wherein we have kind of dead cells which do not have the nucleus. They are kind of just a layer of cells which have lost their nuclei, lost their organelles, and a horny layer which is also called as, it is just a dead layer. We do not say it corneum. Corneum is the one which is present only in skin. Okay, now when we talk about something in the oral mucosa or inside the body, we do not call it directly the corneum. Okay, so let me just give me a second. I think so we do call it corneum. It's the lucidum which is not present. Yeah. Yeah, it's basically the corneum which is present. You know what happens in skin actually? There are two layers. When we come to the horny layer, the upper part, there are two layers. One is the corneum and one is the corneum lucidum. So in lucidum what happens, there are clear cells. It's a clear layer that kind of forms a protective layer on the skin. That lucidum is present only in the skin. And while when we talk about the keratinized epithelium of the gingiva, it doesn't have the lucidum, and yes, its upper layer is called as corneum. I'm sorry about that confusion. Okay? Yeah. Now, but when we talk about, we don't have to make a jingle. This is a very important point or normal concept in this thing. We need to remember it, okay? We'll be talking about these cells everywhere in oral histology. So it's the basement membrane, then we have the basal layer, then we have the prickle cells or the spinous layers. Why we call it the prickle cell or spinous layer, we'll be talking about it. No audio? Others, are you able to hear me? Kamal, you get me now? Kamal's not getting audio, I think. Okay, so when we talk about this layer, I was talking about this as the spinous or the prickle layer. Now what happens is, the spinous or the prickle layer is wherein the cells are attached to each other, but they are trying to be pulled apart because of the extracellular material is pushing them apart. So that is why they get a shape like a star. That is why they are like a prick. That is why this layer is called as the prickle cell layer. After that we have the granular layer. This granular layer has lot of the prickle cell layer is nothing but you have the cells are attached with desmosomes, and whenever there is an extracellular material pulling these cells apart, what will happen? They will remain attached at the desmosomes, but because of the pulling, they will assume a kind of a star shape. Okay, that is why this particular layer is called as prickle cell layer. That prickling happens, that shape of the cell becomes like a little bit prickled, not exactly round. It is like this. Okay, that is why it is called as prickle cell layer, because this particular stage the cells produce a lot of extracellular material. After that we have a granular layer. We will be talking about what this granular layer contains in gingiva in our later slides. Okay, after that we have the horny layer where the granules are lost and they form this keratin. A lot of keratin in the cells. So these cells are filled with keratin. Kamal, your speed is not good. That's why not getting audio and not getting video. Okay. So it's all about the bandwidth. Look at the bandwidth column over there. So work out with your bandwidth so that you'll be able to see the video and this thing. Okay. Hopefully it's not a common problem, because I do see my bandwidth full. Yeah, Karan, this particular stratum lucidum will definitely be covered in your histology slide. As I told, corneum, right? Corneum may cells contain these keratin. So these particular cells of stratum corneum, they are filled with keratin. But in skin, what happens? There are some cells which we, when see it histologically, they appear clear. And they are totally the dead cell forming the line, a protective line devoid of content. Okay, that particular one protective, extra protective layer that we have in skin is called as stratum lucidum. Okay, yeah. Now, but when we compare the keratinized to the non-keratinized gingiva, now look at the non-keratinized version. In non-keratinized version, we have the basal layer, we have the prickle layer, but between prickle and the surface is not corneum. The surface layer is not losing its nucleus. It is having a nucleus. It is not dead. It is not just the squamate. It is a total cell, although it is a little flattened, but it is not dead. Okay, in non-keratinized. There is no stratum granulosum in non-keratinized. Okay, so we have the basal layer, we have the prickle layer, same as in this one. But instead of granular layer, we have an intermediate layer, and then we have the surface layer. We do not call it stratum corneum. Okay, so that's the difference between keratinized and the non-keratinized epithelium. Get the point? Are you all clear with it? Okay, now when we actually go into the non-keratinocytes, now all the layers we talk about keratinocytes are these layers, all the basal, spinosome, everything is made up of the cells, the epithelial cells, which are called as keratinocytes. Any other cells other than these keratinocytes which are present in these layers are called as non-keratinocytes. In that we have the melanocytes. Now melanocytes are present in the basal and the spinous layer. Now you all must be able to imagine which is the layer where we have the melanocytes. It is the lower two layers where we have the melanocytes, and they synthesize melanin. So melanocytes synthesize melanin. This melanin can be eaten by the cells which are called as melanophores. Okay, so if you have your keynote, just scribble down the word melanophores. Melanophores are nothing but the cells which can actually phagocytose the melanin and store this melanin pigments in them. Okay, then we have the Merkel cells. Are you guys with me? Are you all able to hear me? Okay, I think so you can just keep Y and enter. That would actually make me know that you all know the things. Otherwise what happens is I somehow feel that it's not clear, and I may tend to over explain the things which actually is not needed, isn't it? So it may just take in a little bit more extra time than it is required. Okay, so hope you get melanocytes. Then we have the Merkel cells. Merkel cells are again in the deeper layers of epithelium, and they are connected to other cells. These are the tactile cells. They are the modified nerve cells, you can say, and they have the tactile perceptions. Now these Merkel cells are attached to the other cells as well, and they sense the pressure. Okay, then we have the Langerhans cells. Langerhans cells are basically the protective, phagocytic cells. These are the dendritic cells which are located in all suprabasal layer. Now look at the point here. This is the only cell we are talking about which is not present in the lower. It is present in the upper layers. So your Langerhans cells are not present in the basal layer. It is present in the upper layers. Okay. So remember the positions. They are the important source of MCQs. Okay. Now we'll be talking about the histological classification of you tell me the difference between okay, Shubhrajit and others, did you understand what I explained to you in this particular thing? Everybody of you understood? Okay, now can you all tell me which particular layer is missing in non-keratinocytes, non-keratinized epithelium? Which particular layer is missing in non-keratinized epithelium? Yes, the squamous and the granular layer. Okay, so basically the only difference between non-keratinized and keratinized epithelium is the upper layer doesn't become keratinized. Okay, as the name says, when we say the upper layer doesn't become keratinized, that means the upper layer cells do not have that extra deposition of keratin in non-keratinized epithelium. This keratin is formed in the granular layer. Okay, so both the granular layer will not have the keratin and the upper layer will not have the keratin. So both the layers are missing in non-keratinized, which have a different term called as the intermediate layer and the surface layer. They are nothing but the flattened cells. They do not form keratin. That's the difference between non-keratinized and keratinized. Get the point? Shubhrajit? Clear? Yeah, so don't make it complicated, it's not complicated. Make it simple. Try to make it simple. You don't have to put in the extra information just to make it complicated. Extra information is needed where it is needed. Okay? Okay, now remember we had actually differentiated our gingiva macroscopically into different parts, the interdental gingiva and all that. When we talk about microscopically, now let me just go in here. And I will actually now draw a cross section of the tooth, and then we'll be talking about the different thing. Okay, now this is the tooth, this is the gingiva. Now when I draw the epithelium, okay, here we'll have the connective tissue. Connective tissue will be covered by the epithelium. Okay, so epithelium will start here. Get the point? So our epithelium, which is here, attached to the tooth. See this? This whole is epithelium, this layer. It is covering the connective connective tissue. This part of the epithelium which is attaching to the tooth is your junctional epithelium. This part of the epithelium which is lining the sulcus, this is the sulcus, right? This part of the epithelium which is lining the sulcus is your sulcular epithelium, and the part of the epithelium which is on the outer side, is your oral epithelium. Okay, so this is your oral epithelium. This inner side is your sulcular epithelium, and this which is attached is your junctional epithelium. Okay, now just think very simply, your marginal gingiva and attached gingiva both on the outer sides are covered by oral epithelium, isn't it? The outer everything is the oral epithelium. Only the inner side, is your sulcular epithelium. Hopefully if you understand that, then we'll go and see the characteristic and the difference between the epitheliums. Now the oral epithelium, it covers the crest and the outer surface of the marginal gingiva and the surface of the attached gingiva. Now when we talk about the thickness of this oral epithelium, it is 0.2 to 0.3 millimeters. Every value is important. Okay. Now keratinization, the outer epithelium is keratinized. You understand the difference between the keratinized? This keratin actually makes it more resistant to the stress. This outer epithelium is keratinized. Then we have the layers. Obviously, if it is a keratinized epithelium, the layers are stratum basale, we already discussed, spinosum, granulosum and corneum. Isn't it? Because it is keratinized. The uppermost layer of spinosum will contain keratinosomes or odd-length bodies. Now we'll be talking about these odd-length bodies. Okay, these odd-length bodies are present only in the oral epithelium, the keratinized epithelium. Okay, and they are present in the upper part of the spinous layer. So we had our basal layer, then we had the spinous layer, and before the granular layer here in the upper part of the spinous layer, here we are having the keratinosomes or the odd-length bodies. Okay, then we have certain kinds of keratins. Now before I say what keratins are, okay, you have to understand that keratins are nothing but a kind of proteins. Okay. There are various kinds of these proteins that give the characteristic to the epithelium, and their numbering system is opposite. So if I say a number having low number, for example, if I say K1, just logged in, Pankaj, it may take a while before it loads. You have the sound now? All of you have the sounds? Others? Okay. Yeah. So the number K1, the K1 tells that it has a high molecular weight, while higher number tells that it has a low molecular weight. So numbering system of keratins is opposite of its molecular weight. Okay. So now there are various types of these keratins. There are many types of these keratins based on their properties. So you will notice that the keratin, some keratins are present in the epithelium which are highly differentiated. Some keratins are present in the epithelium which are very immature, not differentiated. Some keratins are present in non-keratinized epithelium, while some keratins are present in keratinized epithelium. Okay, they are just the proteins. So they give a typical characteristic to the epithelium. So herein we have to see which keratins are present in which kind of epithelium, and let me tell you, gingiva behaves erratically. Okay, so it will have all the exceptions there. So there are a lot of key points to be made here. You need to have your stickies ready. There are many unexplained things happening here. Okay, So when we talk about oral epithelium, which is a keratinized epithelium covering the outer side, it has K1, K2, K10 and K12, which are the characteristic of differentiated epithelium. Okay, hope you understand what term differentiated it. That means the matured epithelium, okay, which has the specialized cells. K6 and K16 are the characteristic of highly proliferative epithelium which are present in the oral epithelium. Now we all know that gingiva has a high turnover rate, so it has K6 and K16. Okay, K5 and K14 are stratification specific. That means the epithelium that has layers will have K5 and K14. So oral epithelium is stratified and it has K5 and K14. Okay, and K19 is seen only in parakeratinized. Okay, guys, I'll be running out of time. There would be 5 minutes left for this class to end. Okay, so probably wherever I get a pop-up next, we'll just wait for the next part. Right now you don't need a mnemonic here. It's 1, 2, 3, 4. Believe, trust your brain, please. Make stickies, okay? These mnemonics, there is a limit till you make mnemonics. Otherwise, it becomes too confusing if you make too many of mnemonics. Keep the mnemonics for pharmacology. Keep it for microbiology, okay? We'll be talking about mnemonic as much as possible. Now, it's simple. It's 1, 2, 10, and 12. We'll discuss it. 6 and 16, it's so rhyming. 6 and 16, it's highly proliferative. 5 and 14 is stratification specific, and 19. What can we make a mnemonic for numbers? Isn't it? So just relax, relax and think that it will not be so difficult and you will remember it, and you will remember it. Don't worry. Okay? You already do remember it right now, don't you? If I just switch the slide, you will remember it. Okay? Now let's come to the junctional epithelium. We'll compare the junctional epithelium. Don't worry, Shubhajit, just relax, and more you relax with reading such memory-based things, more you'll be able to recollect. Just make stickies, segregate it, make nice charts with your own handwriting. You will be able to do it. Okay? Now remember the junctional epithelium, the position of the junctional epithelium, this one. Okay? The junctional epithelium is a band-like tissue that surrounds the tooth. So it is the one that is attaching the gingiva to the tooth. It has 10 to 20 cell layer thick. This point is important, MCQ. Just a fact, it is 10 to 20 cell layer thick. The length is 0.25 to 1.35 millimeters. I understand this. It will have a lot of values and you have to remember all those values, okay? Every value has been an MCQ. Now look at this thing. Your junctional epithelium doesn't have the typical layer of epithelium. It has only two layers, the basal layer and suprabasal. Okay? It has only two layers. I'll be talking about, when we go to the junctional epithelium in detail, I'll be talking about these two layers more. Okay? It does not have odd-land bodies. The keratinosomes which we saw in oral epithelium, junctional epithelium does not have it. Right? Then, when we talk about the keratin, it has peculiar keratins which do not match its characteristic. Okay? So, all of you have your keynotes. Okay? Now, I want you all, my class is ending, I want you all to analyze these differences, and I'll talk about these differences in my next part, and we'll try to analyze why we have these differences. Okay? So just analyze these differences till our next part. Just see what is different. Right? So I'll be seeing you in the next session in around 5 minutes or so. Okay? Yeah. So you'll have to log in again. Take care. Bye-bye.